Okay, we are back. Like it or not. Quick, hit fast forward to next video. This is your last chance. Spoiler alert. So we're going to answer. This is Mark Tommerdahl, Bob Dennis, Grumpy Scientist.
Attempt to answer
questions from our video number Grumpy Science 102.
Yeah.
From one of our favorite readers.
He's actually a neuroradiologist, I believe.
He's a neuroradiologist, so we got to be really careful so we don't sound stupid.
But he's also he's also a part-time farmer, so he's got his own tractor. So you know, we'll try to take a stab at his
famously, I believe, in a recent video, he discussed the use of a beer holder on his tractor.
But basically, one of his questions actually had to do with differences between young and old. And I'm going to segue into questions that I've gotten several times from other viewers about, how are normative scores on the brain gauge different for young viewers than they are for old viewers, or for normative values, and why? Okay, so
no product placement.
Yeah, no product placement. So basically, think of it this way: you got synaptic density, or cortical density, is sort of at a peak at around 5 years old in a normally developing child.
People have heard about this, right? You start with a lot of neurons and a lot of synapses, and then there's a die-off, and that die-off is thoroughly essential for.
Dang, that was part of the question I knew the answer to.
Oh, but yeah, I'm sorry, I stole your thunder. Go, bad. That must have been Jedediah.
Okay,
stop it. I need an exorcism.
So yeah, you got all these things, and they're packed together. So pack. They're packed really densely, and everybody has heard the old wives' tale or whatever. It's like, oh, you lose neurons from day one. It's like, well, you're not exactly losing neurons, and you're not exactly not growing them back. But you are making more connections, and basically cell pruning happens, and the other thing is cell growth happens, and you have little buds on each neuron. They look like little buds on trees.
Dendritic buds, I think they're called, right?
They're dendritic spines, and they have little. They look like little bitty tree buds, and those things grow, and basically they also have to do with memory, not just. They look like little mushrooms, but the size and shape of them, the dimensions of them, and everything, they all fit into what's called plasticity. And basically, you have cell pruning, where you lose cells, but you gain connections, and you get more and more connections over the course of a lifetime. And what prompts those connections is probably learning and plasticity. And when you quit learning and being plastic, well, I guess you dry up, and that's the degenerative side of things. But you have normative scores for kids around 8 to 12. That's where we have the most data, or in some cases twice as high as they are for adults. Like TOJ, for example.
Where high is not good.
High is not good.
Bad.
So temporal order judgment normal score is in the 20 to 30 millisecond range. That's if you give one pulse. You give a pulse on one finger and another pulse on the other finger, and you can tell which one came first. And normally you're good between 20 to 30 milliseconds, and most kids, the average score is around 70.
Yeah.
If that kid has ADHD, it's around 110. So they have a little more trouble with that. So what most people don't really think about is ADHD is a developmental disorder, and a developmental disorder doesn't mean that they'll always be slower on that particular score. It means it will take them longer to get to that point. So what we saw in our autism research, another developmental disorder, is that it just takes longer for a lot of the people in certain part of that spectrum to get to normal. And you talk to some researchers who actually look at the postmortem histology, like looking at the brain cells of people in their 40s and 50s and 60s who had autism, and they say, well, the cell pruning actually just took a lot longer. It's just their developmental age is much later. And so one question we get is, are normative scores tied to, like when we type in our age, will we get scores relative to the other people in that age group? Well, the answer is you're going to get normative scores for adults right now. It's going to take a lot of research. And the thing is if you have a 15-year-old child, and anybody who's had kids will know this, some kids develop faster than others, and their scores reflect that. So a 15-year-old girl might score like an adult, and a 15-year-old boy might score like a 12-year-old, or vice versa. It just depends on the developmental speed. Right. It really depends on the individual. So, you know, what a lot of studies do, and a lot of people that do studies, they do what are called, they grade people for the developmental age. And so when they're doing an autism study, for example, they'll say, well, this kid's 8, developmentally he's 6. So now we have to get an age-matched control. Should we talk about controls for experiments?
Why do you run a scientific control? That's a really good question. And in fact, there was a dark time where study sections on the NIH in the mid-90s were talking about, well, let's not spend money on these controls because we already know what the results are going to be. Well, the point of a scientific control is that you do know what the results should be, right, in most cases. So you can tell if your experiment has a systematic error, because if you don't get the results that you should get, or if all of a sudden they change, then that tells you there's something kind of in your control group which isn't supposed to change. It's telling you there's something systematically wrong with your experiment. This is a key feature of experimental method. Is controls.
Exactly. So anyway, we won't talk about controls for. Well, the problem with controls is that they're really a mismatch. And so saying somebody's developmentally age 6 or age 5 when they're really 8 or 9, and then do you match them to somebody who's physically 8 or 6 or 9?
It's a really big, diverse group that's not well studied necessarily.
So the thing we're trying to point out here is how much variability there is in a human study. It just goes from, it's just there's many variables that you can think of.
Editorial comment here is that a lot of scientists stay away from human studies because of this variability, because it makes the study so difficult and they have to have so many people to get valid results. But I would argue just the opposite, which is if you can't manage that study with a diverse group of humans, then you're not doing anything that's relevant for a diverse group of humans. That's my opinion.
Yeah. Well, let's grab another question. After a stroke, there's some plasticity, a lot under the age of 10, but not much at age 60. So how do rewired networks transmit inhibition? So what he's asking about is, like, if you wipe out a whole area of cortex, what happens? And so basically you have, I wonder if I should draw something.
And the argument is that when you're 10, you've got all this presumed neuroplasticity and you recover from it, right? Whereas at 60, things are pretty well hardwired. So if you lose something, you've lost it, which isn't really necessarily true.
There is, and a lot of people don't want to hear about this, but there were a lot of studies done back in the 70s. I was just a youngster then. I was not doing them. But what they did was they were doing what are called plasticity studies. And basically, you know what ocular dominance columns are about?
I do.
Okay, you want to explain what they are?
No, I do not, because I will sound stupid. So please go ahead.
Okay, so ocular dominance columns are in the visual cortex. That's part of the brain that deals with vision. Actually, they're V1 through V18, but we won't go into that. And you have one, let's say you've got a column here. That's a whole bunch of cells, a gazillion cells, 300 to 700 microns in diameter. It's right next door to a column here. That's 300, 700 microns in diameter, and it basically has input from, let's say, right eye, left eye, and they're right next to each other. And then on the next side, so we got right eye, left eye, right eye, left eye. It obviously, basically that what happens. So what would be the obvious experiment to do if you were just going to say, gee, I wonder what would happen if you wiped out the input on half those columns?
Half of those columns that are one specific eye dominant, or just randomly half of those?
Yeah, so what they did was they took a certain animal and they put patches over their eyes.
Oh, right.
So they couldn't see.
So they're basically
for the
like
first 3
every
months.
other column in the cortex is being, its input's being kind of shut off.
So this is the way they did plasticity studies, and they basically shrunk up. And so what happened was when
the columns that correspond to the eye that's covered, it's no longer seeing, they kind of atrophy, right?
They just completely atrophy, very small. They get really tiny anyway, and basically, after 3 months, they take the patch off or whatever and say, okay, now what happens to that column? Well, the column slowly expands back with a little bit of input when the animal's young. And if you do this till the animal's old, then plasticity is gone. So plasticity is one of those things that is age related.
It certainly seems to be.
And the more it's been a huge area of studies, and there's zillions of papers on it.
People are trying to live forever or extend their health span, are doing a lot of things to try to maintain plasticity as long as they can. And there are things you can do, right? There's whole groups of people who aren't necessarily genetically related who remain very mentally alert and everything well into their 80s and 90s and beyond.
And this plasticity where you turn off an area, be either experimentally or through stroke or from phantom limb pain. Basically, people have phantom limb pain because they have a whole area of cortex that is no longer receiving mechanical input. One thing that's been learned is that sensory discrimination tasks actually improve the pain that goes with that, because it's actually providing input. Yeah. And you know what an example of a sensory discrimination task is?
Well, yeah, I do, but I was just thinking mentally, like you're talking about now in the somatosensory cortex, where you have a close association between normal sensation and pain. Which I believe, correct me if I'm wrong, that's the only part of the brain where that's really true, is your somatosensory cortex. Right. So sometimes if you're deficient in the normal sensation, you can actually get pain signals kind of bleeding over, right?
Well, that's right. You have two areas in the somatosensory cortex. What a lot of people don't realize is somatosensory cortex has a lot to do with pain. It's the only sensory modality that has pain inputs.
Yeah, none of the other senses in your brain are really kind of
they're really not integrated with pain.
So vision and hearing and stuff like that are not really associated with pain in the cortex, whereas
you hear stuff that you don't like, but you don't have pain receptors in your ears. So, but basically,
as far as your brain's concerned,
but basically, those two places, they're all wrapped in together in somatosensory cortex, one right next to the other. But if mechanical, and let's say this is the mechanical side that receives mechanical input, that's what, like when you do all the tests on the brain gauge, that's what handles that. But you got all these pain inputs here, and it's all part of a network. If pain is too high, then all of a sudden this area gets suppressed and you can't feel anything.
That's right. So don't forget, we have this lateral inhibition, right? Sort of thing where if you've got a big signal that dominates, we had an earlier talk about this, it tends to suppress adjacent areas on the cortex, that sombrero shape. And what Mark's talking about here is that if you have an overwhelming pain signal, it'll tend to suppress the nearby signals related to normal feeling sensation.
Right. And so that also segues into how long do you treat pain or treat something like that. And basically, if you've got this high input of pain and it's suppressing mechanical input, and you drive it down with something, whether it's like PEMF or drugs or whatever, how long will that stay? And how long will that keep in check? And apparently doing sensory discrimination tasks will have an effect on that.
Yeah, that's the interesting thing. So if you're sort of exercising and stimulating that part of the brain that's next to the pain center, but you're doing it with non-noxious signals in a way that's not eliciting pain, then you can actually start to dominate the conversation, right? With your non-painful signals.
It's called remapping. So
remapping your brain.
So the same thing as the ocular dominance column that gets sewn shut, or the phantom limb pain that gets dominant, or the stroke victim that has cortex that's no longer getting input, or the input is getting there, but that part of the brain is blocked. Basically, things rewire around it, or they rewire, or like the ocular dominance columns get bigger. But basically, it has to do with use. And the more that's used, the more that plasticity functions. And it's sort of an exponential rise. So the more that things happen, the more they build. The more sensory discrimination that you do, the more remapping that happens.
Well, I would like to say, if I may, that I think that working with specific experiments that involve sensory discrimination, and our stuff is one of those, our brain gauge technology is one of those things, you're sort of, in a way, you're exercising that mechanism. Is that correct to say?
That's correct.
Okay. Did you want to say anything else?
Nothing else.
I think we're there.
That's
we're
long
almost
enough.
at our time limit, so thanks for joining us.
You bet.